Hello, this is lecture 11. This lecture and the next 2 or 3, I'm not sure exactly how many, will cover a topic called protein targeting. So we're going to look at protein targeting to the nucleus, to the ER, to the mitochondria and peroxisomes. Protein targeting in general is a really important part of cell biology, and I like to talk about it after we've talked about regulation of protein synthesis because I think it's a logical next thing to talk about. For this reason, let's kind of follow the path that we've been going so far. We've started out talking about proteins and how important those proteins are, how much effort the cell puts into making sure that they're synthesized correctly, that they're folded correctly. As you know, the cell is going to do everything it can to fold it correctly, proteins correctly. If they're not folded correctly, it gets rid of them. Then we kind of said, okay, we know how important proteins are. Let's look at how the cell regulates protein synthesis. And so that's what we've done. That's what we've spent the last little section talking about, the regulation of protein synthesis. Now, let's assume that a cell has regulated protein synthesis. It's made the right amount of the protein at the right time. It's folded that protein properly. And so that protein is perfectly fine and it's ready to go. The next challenge is to think about how that protein gets to its right location or its correct location. In other words, if that's a protein that needs to function in the ER, it has to get to the ER in order to perform its function. If it's a protein that's going to be secreted from the cell, then it has to be secreted. So there has to be a mechanism for ensuring that proteins get to the right location. And this whole concept is called protein targeting these proteins to the right location in the cell. Now, we're going to start out by talking about targeting to the nucleus, and we're going to do that because we just spent some time talking about a bunch of things that happen in the nucleus. But before we actually talk about protein targeting specifically to the nucleus, in this lecture I've got a few slides that deal with protein targeting in general. So we'll look kind of at an overview of protein targeting, and then we'll go back and see specifically how proteins are targeted to the nucleus. So here's a really general way to look at this. Let's imagine that that gene has been transcribed, the mRNA has been processed, the mRNA has been exported from the nucleus out into the cytoplasm. And we know that the next step is going to be translation. Now, I know according to the last lecture that we saw, that may or may not happen right away. But let's assume that it's going to be translated and the protein is going to be made. So those events are going to take place. That translation event is going to take place in the cytosol. Now, that's an important concept because, you know, one way that you could think about this is that the cell could localize its mRNA. In other words, you could take the mRNA for a protein that's going to the mitochondria and take the mRNA to the mitochondria. But what's the problem with that? The problem with that is that you would have to have the protein translation machinery in every organelle. In other words, every organelle would have to have the ability to translate an mRNA. Plus, you would have to have the ability to get that mRNA to the organelle. So what the cell does is something which is much simpler, probably much more efficient, and that is that translation occurs in the cytosol. So the translation machinery is in the cytosol, and then once that protein is made in the cytosol, it's directed to its proper location. Now, if you look at this slide right here, you'll notice that there are 3 colors. There's gated transport in red, transmembrane transport in blue, and vesicular transport in green. So the point is that once a protein is made in the cytosol, to get it to its correct destination, there are 3 different mechanisms for doing that. You'll notice that gated transport only occurs in the nucleus. So we'll talk about that first, and we'll talk about why that is. But I'll tell you, in general, that happens because there are large pores on the nucleus. It's the only organelle that has pores large enough to accommodate these folded proteins. Okay, so we'll spend some time talking about that. You can see that next we've got transmembrane transport. This is going to work for peroxisomes, mitochondria, the ER, and plastids, which are found in plants. We won't be talking about those. So this involves a mechanism in which the protein is actually just moved across the membrane. So there's a transport mechanism to get it from one side of the membrane, the cytosolic side of the membrane, to the luminal side. By the way, before we go on, that's a term that's going to come up a lot. Lumen refers to the inside of an organelle or compartment. Okay, so we need a mechanism to get it to the lumen. That's what we're talking about. In other words, we're talking about moving a protein from the cytosol to the lumen of an organelle. Now, it's going to get a little bit more complicated than that, because sometimes the protein doesn't need to go to the lumen. It needs to be inserted into that organelle's membrane. We'll get to that a little bit later on. But in the case of transmembrane transport, we're actually moving the protein from the cytosol across the membrane to the organelle's lumen. And then the third mechanism you'll see is vesicular transport. In that case, what happens is a protein is actually incorporated into a vesicle. And that vesicle then serves as a transport mechanism to get it from one organelle to another. Now notice in that case, we're not moving the protein from the cytosol to an organelle. We're moving it from one organelle to another. This unit doesn't really, we're not going to focus on vesicular transport. Really, what we're going to be focusing on here is gated transport to the nucleus, and then transport to the mitochondria, ER, and peroxisomes. Because everything in green, the vesicular transport, as I said, is really involved in movement of proteins between organelles. And you'll notice here that all the green arrows only occur after the protein has been taken to the endoplasmic reticulum. So it's only once proteins get to the endoplasmic reticulum that they can be incorporated into vesicles and moved to other locations. So we'll have a whole unit on vesicular trafficking. So again, our objective here is to think primarily about gated transport and transmembrane transport. Now, if you look to the right of this, it's basically showing you the same thing. You've got a blue protein, a green protein, and a red protein, a couple color greens, and a brown protein. So notice that the blue proteins are going to move to the nucleus through nuclear pores. This is going to be your gated transport. And then we've got several here in the middle that are going to various organelles like the chloroplast in plants, mitochondria, peroxisomes, and the ER. This is your transmembrane transport. And then on the bottom, again, we've got vesicles. And notice, as I said, that the transmembrane transport gets it to the ER, but then vesicles form at the ER and take the protein to other locations. We're really just going to focus on this initial event, which is getting it to the ER. So once again, we're going to focus on these two in this unit. Now let's think about why this is a particularly challenging problem for the cell. This is an electron micrograph showing you a basic eukaryotic organelle. We can see the nucleus up here in the top. We can see the nuclear membrane right here. And if you look at the cytosol, you can see that it's just chock full of organelles. You've got mitochondria, peroxisomes, lysosomes, the rough ER, the Golgi. So imagine that a ribosome has translated an mRNA. You've got a twofold challenge here. One is you've got to get that protein to the right location, to the right organelle, but there are so many other possible organelles that it could go to. So you've got to have a mechanism for getting it to the right organelle, and that mechanism has to be very selective so that it doesn't go to the wrong organelle. In other words, you don't want a protein that's going to function in the mitochondria accidentally being taken to the ER. One, you don't know what it's going to do when it gets to the ER, and secondly, that protein then cannot be in the mitochondria doing the job it needs to do. So the crowded environment of the cytosol really makes this protein targeting issue a particular challenge. So in general, what we're going to see is that in order to get a protein to a particular organelle, that protein is going to have a signal on it. That signal that tells it where to go is called the signal sequence. Now, interestingly enough, when this whole hypothesis was first put forward back in the 70s, it came from a lab in which the principal investigator was a guy named Gunther Blobel. Blobel eventually won the Nobel Prize for this work, but here's Blobel's analogy. Blobel said that if you want to get a protein to the right compartment, you need to put some sort of tag on that protein so that it knows where to go. And his analogy was the postal service. And he used zip codes as an example. In other words, if you want to get your letter to a certain city or location, you put the address on there and you put the zip code. And so you can think of these signal sequences kind of like you would a zip code or an address on a letter. It tells that letter where to go. The signal sequence tells the protein where to go. Now the question is, what is the signal sequence? Well, it turns out that the signal sequence is actually part of the protein itself. It's part of the amino acid sequence of the protein. So if we look here for protein import into the nucleus, we'll see that we've got a proline, we've got 5 basic amino acids there, and then we've got valine. So somewhere in a protein that's going to the nucleus, it's going to have this amino acid sequence, or one very close to it, that functions as a signal sequence to tell that protein to go to the nucleus. If it's going to go to the mitochondria, it's going to have a different signal sequence. If it's going to go to the ER, it's going to have yet a different signal sequence. Now let's see if there are any common characteristics with these. The first thing you'll notice is if you look at protein import into the mitochondria and protein import into the ER, this is also true, by the way, of plastids and plants. Notice the presence of the amino terminus there. Excuse me. What that tells you is that in that case, the signal sequence is very near the amino terminus. In other words, for those proteins, here's the protein, here's the C terminus, here's the N terminus. The signal sequence in that case is right here at the N terminus. It's the very first part of the protein that's made. Now we're going to see a little bit later on, that's very important. But you'll also notice that's not always the case. If you look at the nucleus, for example, you can't tell where on the protein it is. So if this were a nuclear protein going to the nucleus, here's the N and here's the C. Our signal sequence could be here, could be here, could be here. You don't know. You got to look at the particular protein and figure out where that signal sequence is. The other thing to notice is that some of these signal sequences are relatively short and some of them are relatively long. You'll also notice that if you look, let's say here for import into the mitochondria, you'll notice that we've got some basic amino acids. We've got arginine, arginine, lysine, arginine, and arginine. And you'll notice that they're spaced apart relatively equally. We're going to find that when it comes to protein targeting to the mitochondria, that spacing is really important. So the point is that for each of these signal sequences, there are unique characteristics that help that signal sequence to function properly depending on the organelle. So we'll talk about those as we get to each of these organelles and the signal sequence. Again, we're going to start out with a nucleus. So here's the thing to notice from the nucleus. If a protein is going to be moved from the cytosol to the nucleus, it's going to have a relatively short signal sequence. And that signal sequence is going to be contained somewhere in the protein, not on the amino terminus, not on the carboxy terminus. Now, there's one other thing I want to say about that. If a signal sequence is on the amino terminus or the carboxy terminus, I forgot to say that. If you look down here at the peroxisomes, this one is actually on the carboxy terminus. Here's another one for return to the ER on the carboxy terminus. So sometimes the signal sequence could be here. Now, if the signal sequence is on the end of the protein, then what that means is that sometimes that signal sequence could get clipped. You could remove it, and you can remove it without really affecting the function of the protein. In other words, sometimes the signal sequence is on the amino terminus or the N terminus. Its job is to get the protein to the right location, and then it gets clipped off. Think about that with regard to proteins that have the signal sequence located somewhere within the protein. That can't happen. In that case, you can't remove the signal sequence, because if you remove the signal sequence, it would be like you're cutting the protein in half. So some signal sequences are going to be removed, and some signal sequences are going to remain as part of the protein forever. In other words, some signal sequences function exclusively as signal sequences. Once they've done their job, they get removed. Other signal sequences function as signal sequences, but then they also function as part of the tertiary final structure of the protein. Now, let's think about how these signal sequences are going to work in general, and to show you how powerful these signal sequences are. Let's imagine that you've got two proteins over here on the left. You've got a green protein, which is going to function in the cytosol. Now notice, if a protein is functioning in the cytosol, there is no signal sequence. That makes perfect sense, right? Because translation occurs in the cytosol. If you want that protein to stay in the cytosol, then you just don't put a signal sequence on it. It's going to stay in the cytosol. In this case, the blue protein has an ER signal sequence, therefore it ends up in the ER. Now, look in the right. You can go to the laboratory and you can manipulate proteins using molecular biology techniques. And in this case, what they did, hypothetically here, is they removed the signal sequence from this ER protein, the blue protein, and they took an ER signal sequence and put it on the green protein. And notice what happens. Notice that without the signal sequence, the blue protein doesn't go into the ER, it stays in the cytosol. And with the signal sequence, the cytosolic protein goes to the ER. Now, what does that tell you? It tells you that that signal sequence is both necessary and sufficient. It's necessary to get the protein to the ER. You know that because if you remove it, your protein doesn't get to the ER. So it's necessary. It's also sufficient because it tells you here that if you've got this cytosolic protein, all you have to do is put the ER signal sequence on there and it's going to go to the ER. So it's sufficient. It doesn't require the ER, it doesn't require the signal sequence plus something else. That's enough. So the point is that these signal sequences are very powerful targeting sequences. Their job is to ensure that that protein gets to the right location. All right, with that kind of as a background, now let's turn our attention to protein targeting to the nucleus. Now remember, we saw on an earlier slide, and we'll see it again in just a second, that this is the only place where protein transport occurs by what's called a gated mechanism, a gated transport mechanism. And if you look at the ER, you can see why. Here's a cartoon of the ER, the nucleus. So what you notice, and what I'm sure you already know, is that the nucleus is a double membraned organelle. In other words, it has an inner, let's change color here, It has an inner nuclear membrane, and then it has an outer nuclear membrane. And that outer nuclear membrane is contiguous with the ER. Okay? Now the reason that's significant when it comes to protein targeting is if you look right here, you can see wherever the two membranes fold back on themselves to create the double membrane, you end up with a pore or an opening. You can see one right here. You can see another one right here. Now, these are relatively large pores. In fact, these pores are large enough to accommodate fully folded proteins. Now, so what we're going to see is we're going to see the mechanism by which proteins move through these pores and get to the lumen of the ER. Before we move on, though, a couple of other things we need to say about the nucleus. We've got a double membrane system, we said. It's continuous with the ER. The membrane is punctuated with these pores, and it's lined with a cytoskeleton, and the cytoskeleton is shown in green here. This green lining on the inside of the nucleus is really important because what we're going to see in just a second is that there are a bunch of proteins that are inserted into those pores that are involved in the transport mechanism. And the cytoskeleton actually helps to keep those pores in place. So, one other thing we need to say before we leave this slide is, how would you target proteins to the nuclear membrane? Let's say we want to get a protein to the lumen of the nucleus. That's what we're going to talk about. But let's say we want to get our protein here in the ER membrane. Well, we're not going to talk about that at all. And the reason we're not going to talk about that is because if you want to get a protein to the nuclear membrane, the first thing you do is you get it to the ER membrane, and then it can migrate. So we're essentially going to ignore protein targeting to the nuclear membrane just simply because it's really secondary to protein targeting to the ER membrane. We're going to focus on protein targeting to the nuclear lumen. Okay. So let's look at the nuclear pore and what's called the nuclear pore complex. So when we talk about a pore, we're just talking about an opening. But it turns out if you look at that opening is got some proteins associated with it. And the proteins that are associated with that opening or inserted into that opening is called the nuclear pore complex. Now, the nuclear pore complex is interesting. If you look at it from the nuclear view, so this is we'll just say it this way. It's a nuclear lumen view. In other words, that's what the nuclear pore complex looks like if you were in the nucleus looking up at the nuclear pore complex. That's what you would see. If you were in the cytosol looking at the nuclear pore complex, that's what you would see. Looks very different. One thing you'll notice though, especially if you go down here to the bottom and you look at the cytosolic view, it's an octameric structure. So in other words, you've got one, 2, 3, 4, 5, 6, 7, 8. You can count any one of these and see that 1, 2, 3, a little bit harder here, 4, 5, 6, 7, 8. So it's an octameric structure. Okay, you can kind of see the same thing up here. It may be a little bit harder, but you've got 1, 2, 3, 4, 5, 6, 7, 8. Okay. Also on the luminal side of the nucleus, what we've got is something that's going to create what's called a nuclear basket. We'll see that on the next slide. But that's what this nuclear pore complex looks like. Here's a model of the nuclear pore complex, or a cartoon. So notice here is where the membrane folds back on itself to give you the pore. And then everything shown in a color here is actually part of the nuclear pore complex that's inserted into that pore or that opening. So again, it's an octameric structure. It's a huge structure. It's about 100 million Daltons. Now, just to give you an idea of how that compares to a regular protein, a typical or your average protein is going to be, let's say, about 70 to 80,000. So we've got 100 million compared to 70 to 80,000. So when we take all the proteins that are part of the nuclear pore complex together, we've got this massive complex of proteins that are going to be responsible for moving proteins from the cytosol to the nucleus. Okay? All these proteins collectively that make up this pore are called porins. As we just saw on the last slide, it's an octagonal structure, an 8-sided structure. You've got 8 long filaments that extend into the cytoplasm. These create what are called the terminal ring. Okay, so here it is. Now remember on the last slide, if you're looking at this from the lumen, remember how I drew the 8 lines representing proteins. 8 of those proteins come together. Well, this is what we were drawing on at that point. There are 8 of these that are going to come together to give you this terminal ring. The whole structure there on the nuclear side is called the nuclear basket. As we said, this is embedded into the membrane by part of the cytoskeleton. Now we'll get to the cytoskeleton later this semester, and we'll see that the cytoskeleton has 3 different types of elements. We have microfilaments, microtubules, and intermediate filaments. And these intermediate filaments, called lamin intermediate filaments, are responsible then for helping to make sure that all these proteins are correctly positioned there in the nuclear pore. Okay, we're not going to go over the function of all these. A couple of more that we need to point out, though, is you have these fibrils, these cytosolic fibrils that extend out into the cytosol. Those are going to be important for kind of helping direct things to the pore. And then the other thing that you'll notice here is you've got these gray squiggly lines that kind of extend into the pore. Those are going to be really, really important, as we'll see. Those are actually going to kind of form the gate, so to speak. Again, here's another way of looking at it. Remember, these are our 8 that extend, 8 fibers that extend into the nucleus to form the nuclear basket. The other thing that you can see here, and the reason for showing this, is this cool slide right here shows you that if you actually look, you can see the nuclear membrane right here. There's where we get our double membrane. Same thing over here. And you can see the nuclear pore complex. You can see it actually embedded there in that nuclear pore with your ring right here, giving you your nuclear basket, and then some cytosolic fibrils sticking out in this direction. And again, we've got these that are sticking out in this direction, and these are going to be really important because these are going to function specifically as the gate. As shown on this slide, this region right here with all of these sticking out in here is a disordered region of channel nucleoporins. Now we'll spend some time talking about those channel nucleoporins. Anyway, those are the characteristic features of the nuclear pore complex. Now you might be asking yourself, I thought we were talking about protein targeting. Why are we talking about the nuclear pore complex? Well, because as you'll see, the nuclear pore complex is the structure in the nuclear pore through which the protein has to move. So the mechanism for getting the protein from the cytosol to the nucleus is going to involve these proteins that we're talking about. Now here's the nuclear lamin, or these lamin intermediate filaments. And these intermediate filaments are really important, as I said, because they serve, it's a cross-linked network of fibers. And this cross-linked network of fibers is holding all these proteins in place. Now, let's focus on those proteins that kind of, the squiggly proteins that we've drawn before that extend out into the opening here. These are key to the way this works. A lot of those other proteins are really structural, but these are really play a really important functional role. Now, remember when we were talking about protein structure, we said that there are regions of a protein that are we refer to as random coil. We talked about how important random coil was. We also said that that region of random coil is intrinsically disordered. Okay, not much order there. So these intrinsically disordered or random coil regions of these nucleoporins stick out into the nucleus, into the pore rather. And they're characterized by the presence of these FG repeats. So you'll have a hydrophilic portion of protein, and then you'll have phenylalanine glycine. Hydrophilic region, phenylalanine glycine. Hydrophilic region, phenylalanine glycine. Now what happens is those phenylalanine glycine repeats are nonpolar. So we know what happens to nonpolar things. They end up associating with each other. So what's going to happen then is these intrinsically disordered regions of the protein that stick out into the nuclear pore opening are actually going to associate with each other. So imagine you've got each one of these dots is one of these FG repeats. Well, now here's another protein. You get the picture, hopefully. So what you're creating there in that opening is not only is it occupied by these proteins, but these proteins are sticking together to create a mesh. Now this mesh is important. Think about a piece of like fine mesh. So you got a piece of fine mesh. You're going to take this mesh and you're going to put it over the top of, let's say, a beaker. And then you're going to pour water in it. Well, the water is going to go right through that mesh. And in fact, depending on how large the openings are in the mesh, you may even have some small molecules that can move through that mesh. And that's exactly what happens here. There are some small molecules that are free to diffuse through that mesh. But large molecules can't get through this mesh. Excuse me. I'm sorry. The pollen gives me fits sometimes. I'm here recording this lecture on a Saturday morning, and for some reason the pollen is problematic more than usual. Anyway, so the large macromolecules cannot enter. They can't easily pass through that mesh. So the proteins that we're interested in getting into the nucleus are not going to be able to just move through that mesh. There's going to have to be some other mechanism for getting them through there. Okay, so small molecules can diffuse through these FG nucleoporins, but larger molecules, the proteins that we're interested in, are not able to move through those nucleoporins. So we got to have a specific active mechanism for doing that. Now, this is just to remind us about what the nuclear import signal looks like. Now, don't worry about nuclear export at this point. Let's just focus on nuclear import. We'll talk about export a little bit later on. Here's the signal sequence. You don't have to memorize these signal sequences. What I want you to know here for the nuclear signal sequence is really two things. One is it's relatively short. Three, it's not on either the amino terminus or the carboxy terminus. Did I say two or three? Well, three characteristics. One, it's short. Two, it's not on the amino terminus or the carboxy terminus. Rather, it's located somewhere on the internal part of the protein. And number three, it's made up essentially of these 5 basic amino acids. Now, to show you how important those amino acids are, we can do an experiment kind of like we did at the beginning, a generic experiment. Here, let's make a more specific experiment. So this is a protein on the left that should go to the nucleus. Doesn't really matter what it is, but you can see that we've got lysine, 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 arginine, and lysine. Now, what's shown up as bright here is the nucleus, because the edge of this cell, we can't see the edge of the cell, but we can imagine the edge of this cell would be something like this. Here's another cell right next to it like this. Okay, so that's a couple of different cells there. And so what's happening here is you can use an antibody to see where this protein is. And what you can see in this case is that the protein is in the nucleus. We could do the same thing over here. We'll just draw where we might suspect the edge of that cell to be. Here's another cell. Here's another cell. Notice that it's in the nucleus. Okay. Now, if you go over here to the right, what they did is, again, they went to the laboratory, used molecular biology techniques to replace the lysine with a threonine. So we're going to replace one of those basic amino acids with another polar amino acid, right? But it's not, it's got that hydroxyl group on it, so it doesn't have that positive charge. And in this case, let's outline the cell here. Here's the edge of the cell. Here's the edge of another cell. Here's the edge of another cell. Okay. And notice that what lights up now is not the nucleus. In fact, the nucleus is kind of dark because none of the protein is in there. Rather, the protein is out in the cytosol. It's out here. And that's because that nuclear localization signal has been altered. So it's not going to function as a nuclear localization signal. Again, it shows you the power of the primary structure, because changing one amino acid here means that this protein is not going to get where it's supposed to get. By the way, while I think about it, the nuclear localization signal is often abbreviated as an NLS. So that's our nuclear localization signal. Now, what's that nuclear localization signal going to do? How's that nuclear localization signal going to be responsible for getting the protein into the nucleus? That's the question we have to ask ourselves. Well, it turns out that in order to get it into the nucleus, there are a number of other proteins that are going to play a role in this. So let's kind of look at these proteins in general, and then we'll go back and describe their function. There's a really important protein called RAN. Okay, and what you're going to see is that RAN is a G protein. Now, we already know how G proteins work, and RAN is going to work like a typical G protein. We've got nuclear transport factor 2. We don't know much about that. We won't have much to say about that. We have important alpha and important beta. So these important alpha and important beta, they're also called karyopherins, are going to form a heterodimeric nuclear import receptor. Okay, so you can think of it kind of like this. Here's the alpha subunit. Here's the beta subunit. Alpha important, beta important. The beta subunit binds to the FG nucleoporins, and the alpha subunit binds to the nuclear localization signal of the protein. And so you can see right there we got a connection now. We've got a protein that has the ability to bind to these FG nucleoporins that occupy the pore, and this same complex has the ability to bind to the nuclear localization signals. So not surprisingly, this important is going to play a really important role. And in fact, it's called the nuclear import receptor. So we'll see how it functions. We're really going to focus on the role of the nuclear import receptor and RAN because they're going to play the major role in getting these proteins to the nucleus. So in this cartoon, basically what you can see is that you've got three proteins, cargo protein 1, 2, and 3. They each have a nuclear localization signal. Now, what they're trying to show you here is that that nuclear localization signal can be a little bit different. We saw on that previous slide that if you changed one amino acid sequence, then it wouldn't function as a nuclear localization signal. Well, that was true for that particular protein. Another protein is going to have a nuclear localization signal that's essentially the same, but it might be a little bit different, but it could still function as a nuclear localization signal. Okay. Each of these nuclear localization signals, though, is going to bind to the nuclear import receptor. Specifically, we said it's going to bind to that alpha subunit. Okay. Now, what it shows on the right here is that sometimes that connection is indirect. We don't need to get bogged down here because it doesn't change the way it works. In this case, you have a cargo protein in which the nuclear localization signal doesn't bind directly to the import receptor. It binds indirectly by binding to this adapter protein. That's really a minor point because either way you slice it, it's attached to that nuclear import receptor. Okay, so now let's think about what's going to happen by looking out in the cytosol. So here's our cargo protein with our nuclear import receptor. We already saw that it's going to bind to the nuclear import receptor shown in blue here. Now that nuclear import receptor is going to interact with those FG nucleoporins through the beta subunit. Okay, notice that it's first going to interact with these cytosolic fibrils. So as these proteins are out in the cytosol, kind of moving around by random motion, they're going to interact with or bump into the cytosolic fibrils. That's going to kind of catch them. By interacting with the cytosolic fibrils, you can think of those as kind of like a collection net. That's going to then concentrate these proteins and allow them then to interact with the FG nucleoporins. So the beta subunit now is going to act with these FG nucleoporins that project out into the pore. The protein is going to move through. Okay. Once it gets inside, it's going to drop off its cargo, and then it's going to go back out. Okay. Now, I've ignored so far on this slide talking about RAN, but RAN plays a really important role here. So let me erase what I've colored and go back now and talk about RAN. Here's what RAN's going to do. The import receptor binds the cargo and it moves through. Okay. Once it moves through, inside the nucleus, there's a RAN GTP. Now, if it's RAN GTP, it's active, right? Because that's the way G proteins work. It turns out that these nuclear import receptors will also bind RAN GTP. When RAN GTP binds to the nuclear import receptor, it induces a conformational change. That conformational change reduces the affinity that the receptor has for the cargo protein. And so the cargo protein is released. Now, RAN can move back out. Once RAN gets back out, it has the, once the receptor gets back out with RAN attached to it, it's going to encounter a RAN gap. You actually can't see the gap on this slide, but you're going to have RAN GTP, RAN GDP. It's going to be, the GTP is going to be converted to GDP, and that's going to dissociate because if it's got RAN GDP, it's no longer active. So it dissociates. Now we have an empty nuclear import receptor, which could do the whole thing again. Okay? So you're going to have RAN GTP in the nucleus, and you're going to have RAN GDP outside the nucleus. So this association, let me just summarize by saying it this way. The association of RAN with the import receptor reduces the affinity that the import receptor has for the cargo protein. When it's out in the cytosol, it's not bound to RAN, and because it's not bound to RAN, it has a high affinity for the cargo protein. Now, before we go on, notice that these arrows are double-headed. We've got them going in both directions. That doesn't make a whole lot of sense since we're trying to get net movement into the nucleus. But as we're going to see in just a second, we're going to look at what accounts for unidirectional movement. But what you can see here is basically the important role that RAN plays. RAN essentially is going to determine whether or not the import receptor has a high or a low affinity for the cargo protein. Now, this is just another way of looking at the same thing. Here's your import receptor on the left. Here's your binding site for your cargo protein right here. Here's your binding site for RAN. And again, what we said is here's your cargo protein bound to the import receptor. When RAN also binds, we're going to induce cargo unloading, and we're going to induce cargo unloading because the cargo is going to be released, and the cargo is going to be released because you can see a change in affinity. Then once again, when the GTP is, when the RAN rather is released, now we've got another empty import receptor, and we could get cargo loading because the nuclear localization signal will now allow it to bind. So in order for this to work, we've got to have a RAN GEF in the nucleus, and we've got to have a RAN gap in the cytosol. This is crucial, right? Because what we said is that we want cargo unloading in the nucleus, and we know that to get cargo unloading, the RAN needs to interact with the nuclear import receptor. And so that means it's going to be in its active form. So we need a RAN GEF to convert it into its active form. In the cytosol, once it gets in the cytosol, we want to change it back to a RAN that doesn't have GDP or that doesn't have GTP on it, so that it will have a high affinity for the cargo protein. So the localization of RAN gap in the cytosol and RAN GEF in the nucleus is absolutely crucial. Now, I really like this slide because it puts everything together and it shows us how it works. So here we go. This is from a different textbook, but I think it's the best figure that I've got on this, so I like to use it. Here's your cargo protein with its nuclear localization signal. There's the nuclear localization signal. It's going to bind to the import receptor, important. Why is it going to bind? It's going to bind because that receptor has a high affinity for the nuclear localization signal. It has a high affinity for the nuclear localization signal because the protein is not bound, doesn't have a GTP bound to it. Doesn't have RAN bound to it, is what I'm trying to say. Okay, now it's going to get in. Now when it gets in, it's going to encounter a RAN GTP. That RAN GTP is going to bind to it. It's going to change the affinity that the receptor has for the cargo molecule. The cargo molecule is going to be released, and now what you have is a complex of important with RAN GTP. That RAN GTP is then going to move out into the cytosol. Once it gets into the cytosol, the RAN is going to encounter the gap. The GTP is going to be converted to GDP, and when it does so, the important receptor will be released. Now what happens to the RAN GDP? The RAN GDP can get back in, maybe through NTF2. And once it gets back in, it'll encounter the GEF and it'll be reactivated and ready to go. Okay, does everybody follow that? Hopefully. Now, we need to ask a really important question. How is unidirectional transport achieved? Now, what do we mean by this? Remember I showed you on the other slide that there's actually a double-headed arrow. It looked like this. So let's think about it like this. Here's your cargo complex. You've got important and you've got your cargo. So it can move this way, but it can also move this way. It can go both ways. In other words, there's nothing really driving it to the inside or making it go to the inside of the nucleus, except for one thing, as we'll see. Likewise, over here, this complex could move in this direction, or it could move in this direction. But in order for this system to work, we need net movement in this direction over here, and we need net movement in this direction over here. In other words, we need unidirectional movement or transport. So how do we get that? Well, let's see how we get it. Notice that what happens with the cargo complex is that the cargo complex is going to move from the cytosol to the nucleus. But what happens as soon as it gets to the nucleus? As soon as it gets to the nucleus, it dissociates. And it dissociates, the cargo dissociates from the receptor because it encounters the GTP. So you're always going to have a high concentration of cargo complex in the cytoplasm and a low concentration of cargo complex in the nucleus. We know that things tend to move down their concentration gradient. And so what happens here is you've got a concentration gradient, and that concentration gradient is high over here and low over here. So the concentration gradient is what drives the movement in that direction. You got the same sort of thing going on over here, because what happens is you've got this complex now, which consists of the important and RAN, but as soon as that, so we got a high concentration over here, but as soon as that gets out into the cytosol, what happens? Because it encounters the gap, this dissociates, and so we're always going to have a low concentration of this out in the cytosol, or we're going to have a low concentration of this out in the cytosol, because as soon as it gets out there, it's going to dissociate. So this concentration gradient goes in the opposite direction. Now there's another way of looking at this, which is important as well. Or another way to say this, those concentration gradients are achieved because GEF is localized to the nucleus and GAP is localized to the cytosol. And because GEF is localized to the nucleus and GAP is localized to the cytosol, it means that you have a concentration difference with RAN. In other words, the RAN that's in the cytosol tends to be in the inactive form. The RAN that's in the nucleus tends to be in the active form. And so really what you can say is this: your localization of GAPs and GEFs ensure that you have RAN GDP in the cytosol and RAN GTP in the nucleus. And the concentration of RAN GDP in the cytosol and RAN GTP in the nucleus is what's responsible for this concentration gradients, the concentration gradient with the important receptor. And so those concentration gradients are going to drive the unidirectional movement. This really just says the same thing. Achieving unidirectional movement of nuclear import is due really to the location of RAN gap and RAN GEF. Okay. Now let's talk about nuclear export for just a second. Some proteins are going to cycle between the nucleus and the cytoplasm. And so if that protein is going to leave the nucleus, it needs a nuclear export signal. Now, I don't want to spend a lot of time on this because if you understand nuclear import, you understand nuclear export. The only thing we have to say here is that the nuclear export receptor is different from the nuclear import receptor. But otherwise, it's going to work exactly the same way, except what's going to happen in the nucleus is you're going to have your cargo protein with its nuclear export signal bound to the exportant in this case and the GTP. So in this case, when GTP is bound to the receptor, it actually has a high affinity for the cargo rather than a low affinity for the cargo. Okay? So in other words, over here, remember with nuclear import, when the receptor is in the nucleus and it has RAN GTP bound to it, that causes the cargo to dissociate because of the low affinity. If you want to export, then you have a high affinity. So we're going to export this out. The RAN GTP is going to be converted to RAN GDP, and now you're going to have a low affinity. Okay, so you're going to get dissociation. This can move back in, and we'll do it all over again. So two big differences here. The mechanism is exactly the same with two big differences. The interaction of a nuclear export signal with exportin occurs with high affinity if the nuclear export if the exportin is bound to RAN GTP. So the affinity is just reversed. And then the other big difference is that when you export the protein, you have a trimeric complex: exportin, RAN GTP, and the cargo. Whereas when you're importing a protein, it's only the importin and the cargo. This just describes in words how you get this unidirectional transport, so I'm not going to elaborate on that. We've already talked about it. I just want to end this by saying this. I like to talk about import into the nucleus at the beginning of this section on protein trafficking for one specific reason. And that is because we just finished a unit where we talked about proteins that are involved in the nucleus. So think about all those proteins that are involved in adding the cap, adding the poly A tail, all those proteins involved in splicing, all the proteins involved in DNA replication and DNA repair. We didn't even talk about those. Think about all the histones. All of those are proteins that function in the nucleus, but they're made in the cytosol. So all those proteins that we've been talking about, all those proteins that play a role in regulation of gene transcription are made in the cytosol. They have a nuclear localization signal, and this, what we just described, is how they get to the nucleus. So that wraps it up for nuclear localization.